session to the videos what I have been taking up on NEET PG entrance. So today's video I'll be mainly focusing upon the NEET MDS PG entrance, and in MDS PG entrance basically about the oral pathology, and in oral pathology slowly we will be covering the entire syllabus, the one-liner MCQs, and I hope from this video you will be getting to learn all the important MCQs what are very frequently asked in the NEET MDS PG entrance exam. So students, starting with, so firstly we'll be beginning with the viral infections, and amongst the viral infections we have the herpes simplex. So the human herpes virus, it is of 8 types. So you can see that the human herpes virus 1, it is also called as the herpes simplex virus 1, and it is called as the oral herpes virus. Next, the human herpes virus 2, that it is also called as the herpes simplex virus 2, and it is responsible for the genital herpes. Moving on to the third, that is the human herpes virus 3, which is called as the varicella zoster virus, human herpes virus 4, which is called as the Epstein-Barr virus, and human herpes virus 5, which is also known as the cytomegalovirus. Now, HHV6, that is, it is related to the etiology of the Langerhans cell histiocytosis. HHV7 is not there, and HHV8 we have, it is responsible, that is the human herpes virus 8, it is responsible for the etiology of the Kaposi sarcoma. So Kaposi sarcoma, when we understand, it is the malignancy of the endothelial cells. Now students, starting with the human herpes virus. So the human herpes virus, when we look at, so the lesions of this human's herpes simplex virus, these lesions are called as cold sores in the oral cavity, and they are also known by the other name in the form, they are also known as fever blisters. Now, once we look at the herpes simplex virus infection, so infections, if it is the first time infection, as we can see, it is called as the primary infection, and if this occurs again, so that is referred to as the secondary herpes simplex virus infection, which is also called as the recurrent herpes. Now moving on, the first time when the infection occurs of the herpes simplex virus, it is called as the primary infection, and it usually occurs in children and young adults. So students, do remember in the NEET MDS PG entrance exam, in oral pathology, when it comes to the QMCQs, very important, if for the first time if the herpes simplex virus infection is occurring, it usually occurs in children and young adults, and mostly in the first place it manifests in the form of herpetic gingivostomatitis, and it is the site what are involved are the gingiva, and in the gingiva basically the attached mucosa. So the first time when the herpes simplex virus infects, this leads to the formation of a painful ulcer, and this painful ulcer is with an erythematous halo. Now, since the first primary infection is occurring, so this is primary infection, the herpes simplex virus, the fever blisters or the cold sores what the patient is having, it heals within a period of 7 to 14 days. That is, maximum of 2 weeks is required for the healing of the primary herpes simplex virus infection, and that too this healing is without scar formation. Now moving on to the next important fact, that is, sometimes the viral particles travel along the fifth cranial nerve. So once they travel along the fifth cranial nerve, that is the trigeminal nerve, so through the trigeminal ganglion, so this is there, it gets in the form, it gets in the latent form, and later on it can get even reactivated. Now the reason for being reactivation of the herpes simplex virus is it can get reactivated by sunlight, by stress, in immunocompromised states, or in the the nutritional deficiency. So, in such cases, this herpes simplex virus can again get reactivated, and this virus particles all travel along the fifth cranial nerve, reaches the trigeminal ganglion, and it remains, the infection remains in the latent form. Now, when we look at the secondary infection, so secondary recurrent herpes causes, basically it leads to the formation of the herpes labialis. Now we move on to the next important point, that the diagnosis of the herpes. So diagnosis of herpes is basically through the Zank cells. So Zank test is done, and in the Zank test we found the multinucleated giant cells are there. So we cannot differentiate on the basis of giant cells or the Zank test. We cannot differentiate between the herpes simplex virus 1 and HSV2. And it also cannot differentiate between the herpes simplex virus and the varicella zoster virus. So now next, the diagnosis is the by the cytopathic effects, because the virus cell has undergone some changes inside its cytoplasm. So certain cytopathic changes can be seen under the microscope, and the first very important one, it is the ballooning degeneration. So the ballooning degeneration is seen in the cell is enlarged. The next, what we can look in the microscope for this herpes simplex virus, it is the nuclear molding. So the what is occurring is, that is the margination of the chromatin occurs. So if this is the nucleus, so whatever the chromatin material are there, that gets towards the margin of the nucleus, and so this is called as the nuclear molding. The next very important pathognomic feature of the herpes simplex virus, it is the lip shoots body. So do students remember, so this lip shoots body, it is actually the intranuclear inclusion bodies what are seen in herpes simplex infection. And apart from this, when it comes to the diagnosis, we can have certain special stains. So these special stains, for example, the Wright stains, the Giemsa stains, the Pap stains, and the fluorescent stain, most importantly the rhodamine stain, is specific for herpes simplex virus diagnosis. Now, next most sensitive and the most specific for diagnosis of the herpes simplex virus infection, it is the polymerase chain reaction. So it is the PCR which is helps in the accurate diagnosis, and it is the most sensitive and the most specific for the diagnosis of the herpes simplex virus infection. So students and viewers, this was a very short discussion where I have discussed in simple terms about the viral infections, particularly the herpes simplex virus, the 8 types of it, and in herpes simplex virus, what is the primary herpes simplex virus infection, how it occurs in what is secondary herpes simplex virus, and how we do the diagnosis. So basically in the NEET MDS PG entrance exams, when you are preparing from oral pathology, so this herpes simplex virus, the points what I have discussed will actually help you in solving the multiple choice questions what are frequently asked. So students and viewers, stay connected. The next NEET PG entrance exams, these short notes, this preparation, this my short notes will certainly help you. So students and viewers, thank you for watching. And then stay connected for the next video, and in the next video I'll be taking up the oral pathology topic further.